Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, Agena Bioscience, I'd like to welcome you to Evaluating Drug-Drug Interactions, Implications of Phenoconversions. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenters for today. First is David F. Kisor, Pharm.D, FCP, Professor of Pharmaceutical Sciences, Director of Pharmacogenomics Education at Manchester University. And our second presenter is Dr. Robin Everts, PhD, Staff Scientist, Global Pharmacogenetics and Clinical Genetics in the Department of Scientific Affairs at Agena Bioscience. Welcome, Dr. Kisor. The presenter ball is yours.
Hello, everyone. Thanks for joining in the topic on pharmacogenomics. You know, we really want to think about what this adds to the overall clinical decision-making that we have in drug therapy. And certainly when we talk about patients, we think about not only the patient's medications being efficacious or causing adverse events, but what are the interactions of potential drugs? So drug-drug interactions are main import, really, in many cases, especially in the older folks who may be taking more medications. And there's implications of phenoconversion, and that's why I'd like to go ahead and discuss. Okay, so when we talk about phenoconversion, we're going to talk about it in the context of pharmacokinetics and really the time course of drug absorption, distribution, metabolism, and excretion. But we're going to talk about specifically two different components of how a drug is eliminated, if you will, from the body. Enzymatic changes in a chemical structure is one. So on this slide on the left-hand side, you see examples with CYP2C19, cytochrome P452C19, and also thiopurine methyltransferase. And a large majority, the vast majority of drugs really are administered in their active form. Some drugs are prodrugs. They're not active. You have to convert them to the active form. But today we're going to talk mostly about the active form. So here, an active drug being converted by CYP2C19 to a metabolite. Same thing with thiopurine methyltransferase. And we see the genes that produce the gene products, the proteins. The other component that we're going to talk about are transporters. And again, drugs moving out of the body. They can also move into the body by transporters, influx transporters or efflux transporters. And I have a couple of examples on the right side of the slide, SLCO1B1, which is the gene that produces the organic anion transporter, the protein 1B1, and then ABCB1, which produces the very researched P-glycoprotein efflux transporter. So in that context, we want to think about different categories of individuals' ability to metabolize medications. And we have standard nomenclature now from the phenotype, how an individual is going to express their metabolism. And we have these 5 different phenotype categories for metabolism: the ultra-rapid, rapid, normal, intermediate, and poor, going from most efficient elimination, the ultra-rapid, to the least efficient, the poor metabolizer. We also have adopted now some transporter common terminology for function: increased function, normal, decreased, and poor function. So in this context, an individual may be put in a category based simply on their genetics, notwithstanding potential drug interactions and how this can affect exposure to medication. And that's the context I'd like to really talk about this in, is really exposure. We're going to talk about area under the curve with some context to alterations in exposure. So this slide shows you 5 different phenotype curves. You can see the ultra-rapid has the lowest area under the curve on the far right on the table, and poor metabolizer has the highest area under the curve, the highest exposure. So let's look at this with a little bit more depth. So if we look at the ultra-rapid metabolizer, what are the consequences of somebody who is very efficient at eliminating the active compound? Well, we can think about this in terms of drug efficacy. And we have some data, albeit a little bit older here, but this is data that shows the percent response to a first medication used for a certain indication or across different drug classes. So we are constantly fighting this battle to get the person on the right medication early on in therapy. And it's one that we oftentimes lose in our world of trial and error therapeutics. And so we have to give the medication. If it doesn't work, give a second medication or a third. And as an example of antidepressants, I recently saw that number may approach 50%. But think about the potential consequences. Somebody with major depressive disorder who receives a medication, doesn't work the first time, and now they need a second medication. There could be some dire consequences. So we need to think about those kind of aspects and drug exposure. So the ultra-rapid individual, ultra-rapid metabolizer, really, when given an active compound, may have the least exposure to the medication and not benefit from it as desired. So let's now look at the other end of the spectrum and talk about the poor metabolizer. So if you were to color in below that blue curve on the slide, below the bottom of the rectangles there only to point out the poor metabolizer exposure, the highest concentrations, but the biggest area of the curve. What are the consequences of a person who has inefficient metabolism and is a poor metabolizer, and how can that impact that individual? Well, here's an example of warfarin relative to CYP2C9. And I have this very basic general pharmacogenetic equation where AUC equals the bioavailability, the drug that reaches systemic circulation, times the dose over clearance. And I put that on here only to show that AUC and clearance are inversely proportional. So clearance is a term that we talk about with elimination of drug from the body, if you will, in a general sense. And I wanted to bring that up because on this slide we have data from a CYP2C9 genotype study and looking at S-warfarin. And you see the 3-3 and the 3-2 genotypes on the x-axis. We've adopted a star nomenclature or asterisk nomenclature, and it really should be star 3, or star 3, star 2. The point is they have the lowest clearance, and therefore they're going to have the highest area under the curve. So poor metabolizers, low clearance, high area of the curve, high exposure. If you look at the right side of this slide, you'll see different star allele diplotypes. And so you see the star 2, star 3, and star 3, star 3. Those are poor metabolizers. And the star 3 form of CYP2C9 has much greater decreased function and activity than the star 2. At any rate, you can see that individuals that are poor metabolizers based on their star allele diplotype and their related phenotypes, they're going to have trouble getting that drug out of the body. The drug is going to have higher concentrations and exposure. And we see also this occurring in the transporter example. So here's a transporter, the gene again, which I mentioned earlier, SLCO1B1. And in the coding region at 521, C replaces T, and the C is a risk allele, a risk form that increases exposure because there's decreased function of the transporter. And so the drug cannot be moved into the liver as readily as it would be. And in the liver is where it gets metabolized, but also where it elicits its effect. So now we've got the form that decreases that transporter. And if you're homozygous and you have C from one parent and C from the other, then you're considered a poor function individual, and that's what this slide is pointing out. And in this image, you see that simvastatin has an increase in area under the curve, the exposure of 221% in the poor function individual. So again, less drug going into the liver, less drug being metabolized, maybe less of an effect where the drug elicits its pharmacologic action. But if it's not going in the liver, it's free to go elsewhere. And one other place it goes is skeletal muscle, and here's where we see the myopathies that are associated with some of the statins. So we have two examples of genetics that are on the two different platforms, one being drug metabolism and the other one being transport. And exposure is the key, the key to considering inefficacy or adverse drug events. And speaking of adverse drug events, this data is from the FDA Adverse Event Reporting System. Now, the first thing I want to point out is that if you go back to 1987 through Q1 of 2020, The increase you see in 2010 and beyond, really this is not just related to more adverse events. There may be, we have older population, so older population has decreased physiology, and because of that, they have greater exposure. Their liver, their kidneys, drug doesn't get cleared by those organs as readily based on a physiology issue. But then you have pathophysiology added on top of that, and so you have greater exposure. But this is also due to other factors not related to really to physiology or pathophysiology or drug interactions, better reporting systems and the like. But my point is that we are seeing a large number of serious adverse events and deaths. So you've got the situation where greater exposure may be explaining some of it. So when I think about patients that I deal with, and I have an expectation that they would respond to a standard dose of a medication and they don't, if they don't respond and they're inefficacious or the drug's inefficacious, then I'm thinking about, okay, what is their metabolizer status? And of course, we haven't had the technology to really get to this as readily as we do now with some of the newer platforms that are out there. And on the other end, if somebody has an adverse drug reaction, I'm thinking, okay, what is causing this? There's physiology, there's pathophysiology. Because of that pathophysiology, they're receiving a medication, maybe multiple medications for multiple pathophysiologies, and now I'm thinking about drug-drug interactions. And then I have to start putting together what's going on with drug-drug interactions relative to the patient's genetics. So I want you to get this picture of exposure. The gray bars that are vertical in the middle would be the normal exposure. The yellow bars project a decreased exposure. That would be the rapid or ultra-rapid metabolizer individual receiving an active drug. And then the green bars really represent that intermediate or poor metabolizer group that is receiving active drug, but they're not readily able to eliminate it. They have greater exposure. So I think this is a way to think about this. And there's other parts that make up the area under the curve. It's not just higher concentrations early on. It's also the fact that there's slower and faster metabolism. So when we look at PGX and drug exposure, I have a couple of examples here, and looking at the percent change in area under the curve, again, as this exposure measure. So if you look at CYP2C9, the gene and the gene product, and the analgesic celecoxib, we see that intermediate metabolizers have an increased exposure of 43%, and poor metabolizers 233%. Again, this is just the inherent difference in individuals that would explain, or at least in part explain, these changes in exposure. So it's no wonder when you look at this kind of data that we have different responses for different individuals. The middle row has CYP2C19, the gene and the product, and now clopidogrel, which is often an example in pharmacogenomics. It's one of the earlier drugs where we applied pharmacogenomics. I can remember the trade name product being advertised on TV, and at some point there was a voiceover at the end of the commercial saying that genetic testing may be ordered by your treating physician or something along those lines. And that's where we first recognized that with clopidogrel, which is a prodrug, and again, it's not an active drug, your body has to convert it to the active form. We realized that individuals who have decreased metabolic capacity, the intermediate poor metabolizers, don't convert all of clopidogrel to its active form. And when we look at the exposure to the parent compound, we see the intermediate metabolizer having a 67% increase in exposure, and a more poor metabolizer, having a 194% increase. So that's the increase in the parent compound, which means there's less being converted to the active compound. So the third example is metoprolol, and we'll have a patient case coming up with this drug and the consequences of the genetic influence, but also a drug interaction. So CYP2D6 being the gene producing the CYP2D6 enzyme, and metoprolol has increased exposure in intermediate metabolizers of almost 50%, 48% In poor metabolizers, 488% as an example. And then if we go to the other end, the very, very efficient metabolizer, that ultra-rapid metabolizer, they have a decreased exposure. And that's a 62% decrease in exposure. So depending on the form of the gene, we can have increased or decreased exposure. And so it can get a little bit complicated. But all three of these examples are really telling us that people are different, right? And why we would expect people to have inherent differences in their response to medication. Now, I don't want to put the response to medication solely on pharmacokinetics, because certainly there's the pharmacodynamic aspect, right? A direct interaction of a drug with a receptor eliciting some response. So we have pharmacokinetic changes and we have pharmacodynamic changes, both with underlying genetic components. So when we think about these associations, the FDA recently, in February, a couple months ago, they produced their first public tables, including relationships between genes and drug exposure. And this was a real big step, because up until this point, the FDA had provided a table of pharmacogenomic biomarkers in drug labeling. And that was helpful to a lot of an extent, I guess, when you think about looking at the drug labels. But there's not a lot of meat, if you will, in the drug labels for the vast majority of medications. And so when we're talking about getting this pharmacogenetic associations from the FDA, it really wasn't there by and large until February 20th. And then February 25th, they updated it really quickly with a change. But what they showed were three different tables in essence. It says that they table, but there's really three different parts. And the first part of the table really was related to that first row in light gray, where the pharmacogenetic associations were related to management recommendations, so therapeutics, right? And they had 51 total gene-drug interactions. And in some cases, there was an increase in exposure, in some cases a decrease, again, depending on the form of the gene. And you can see the genes involved on the far right. The second table really was related to safety. And of course, that's in my view, the big issue with the FDA. And they want to be very careful when they make statements to the public. And so they're very conservative in their safety statements. That's really good to see now that for the first time we have information provided by the FDA related to safety and, again, drug exposure, either increased or decreased exposure. And again, this is across different kinds of drugs. And what I mean by that is active drugs or prodrugs. So again, active drug, it's already active in the body, and the metabolizer status really talks about getting that drug out of the body. Where a prodrug is not active, and the metabolizer status really talks about converting that drug to the active form. And so you can see, for instance, in the far right, in the middle gray column, we have SLCO1B1, and that was a relationship really born out of the potential for that C form, or the heterozygous or homozygous C genotype, resulting in myopathy. That's kind of an example for you. And then the third component of the table, which is really a separate table when you look at the website, and the link is at the bottom of the slide. And you should have access to this, so you'll be able to look at it if you haven't already, likely have. At any rate, the third table was really related just to PK, just to the concentration data. Does an individual have increased or decreased exposure based on some drug-gene interactions? And there were 38 drugs listed. And again, depending on whether or not it was a poor or intermediate metabolizer, again, that's decreased metabolism, then they'd have greater exposure to an active drug. If it was decreased metabolism relative to a prodrug, they would have less exposure to the active drug. So we have to put that in context. But the point is, the FDA really helped us out here by putting some of these pharmacogenetic associations out there for broader dissemination. And when we talk about drug-gene interactions and dissemination, In the U.S. and worldwide, really, the Clinical Pharmacogenetics Implementation Consortium, CPIC, has been working since really 2009 to 2011 roughly to provide evidence-based pharmacogenomics guidelines. So this organization, I think it's over 400 individuals from around the world. They work along with the pharmacogenomics knowledge base to vet the literature to a great extent. They really dig in as far as absolutely possible to get all the information. And they work over years to put together guidelines and then they update guidelines. So currently there's 24 CPIC guidelines. You see some examples in the left side of this table with the green box around it. And so you see some of these are pharmacokinetics, the HLA-Bs are pharmacodynamics. So we have these guidelines. And we also have another group. There's actually 3 or 4 other groups, but the Dutch Pharmacogenetic Working Group on the right side of this slide also has guidelines. And we're going to actually look at some information from that group. What's really important is CPIC played a major role in working with the Centers for Medicare and Medicaid Services to really have a result in a new coverage determination, which will go into effect July 26th. The link to that coverage determination is on this slide, and that really is going to open up the use of pharmacogenomics to a greater degree. The private insurers have started to move on this also, but with CMS moving, it's really going to help drive things. And it's really a broader coverage. It's not as specific as some of the previous coverages like CYP2C19 and clopidogrel as a specific coverage. So really good news. And CPIC played a major role in the coverage being determined. So I would encourage you to look at that coverage and read the document. You can see the role that CPIC played in providing that evidence-based medicine to help this happen. Okay, at this point, what I'd like to do is kind of put this exposure idea together. We've already talked about the underlying genetic basis and the fact that there may be patients who are on multiple medications, and now you have a drug-drug interaction and a drug-gene interaction. And let me use this as an illustration of what could happen. So Robert's a 57-year-old African-American male with heart failure. He's been getting the beta-adrenergic blocker metoprolol, 100 milligrams once daily for his heart failure treatment. But he's also been dealing with depression, and so he started on paroxetine, one of the selective serotonin reuptake inhibitors. 3 days after being on that medication, he was seen at the emergency department, and he had suffered a broken arm. He had fallen, he was getting up, he got dizzy, and he fell. And so likely orthostatic hypotension. And as part of his discharge process, they got into a discussion about a medication. So he explained to the pharmacist at the time that was doing the medication review what he explained was he was a techie, at least as much of a techie as he could be with getting some help from his grandson. So he had some information from direct-to-consumer information that was provided. And of course, there's direct-to-consumer information is provided by different companies, and there's different genes and different gene variants that are tested, and it really muddies the water, in my opinion. But at any rate, in this case, Robert explains these 3 different kind of results that were explained to him by his grandson. And we're going to look right at the CYP2D6 and look at the star nomenclature diplotype. And so he's a star 4, star 17, which works out to mean that categorically, from a phenotype status, he's an intermediate metabolizer. So let's forget about the diplotype and genotype and focus on just that intermediate metabolizer in the context of drug exposure. Metoprolol is given in the active form, so the intermediate metabolizer is going to have greater exposure just based on their genetics. And what that plays out in is a higher area under the curve, right? So this exposure is there for higher concentration and a longer period of time. So if we were expecting Robert to have a normal exposure, And I want to point out that these are not exact AUCs related to metoprolol, these for illustrative purposes. We would expect an AUC of around 20, let's say. However, because of the genetics, Robert really is an intermediate metabolizer, and therefore his exposure is roughly double, around 40 micrograms per milliliter times hour, or milligram per liter times hour, same thing. So the point is, just based on the inherent genetics, Robert's already got increased exposure, but that's not the end of the story. So what is the implication now from a population kind of idea? Like, where does Robert fit? How many people are intermediate metabolizers? Well, according to information from CPIC, about 1 to 2% are ultra-rapid metabolizers. The large majority are normal, but 2 to 11% can be intermediate metabolizers, and then 5 to 10% poor metabolizers. So if we take the intermediate and poor metabolizers, we could be talking about 20% of individuals roughly. That's 1 of every 5 people. So we really need to be thinking really out of the chute that exposure is likely to be greater for a significant number of individuals. Now, what's the consequence of adding paroxetine now? So we had a drug-gene interaction, intermediate metabolizer, but now we're adding paroxetine to metoprolol. Well, here we've got a drug-drug interaction. Paroxetine is a potent 2D6 inhibitor. So where the decreased metabolism was in place based on genetics, now we have even further decreased metabolism. But I want to interject. We wouldn't know this unless we had the patient's genetic profile. So I would argue that until we fully implement as a standard pharmacogenetic testing across the board, interpretation of a patient's medication list cannot be optimal. Now, I say that in the context of the real optimization is looking at the patient, right? Following the patient, not looking at some number or some AUC or some concentration. Sure, and I get that, but we don't want to be chasing our tail to do that, right? We start the patient on a medication, they're an intermediate metabolizer, they have greater exposure. Now we add a drug on board that's going to further decrease their exposure, right? So now we've got this drug-drug interaction, and what's going to change for this patient? Well, we saw what happened to the patient. They had orthostatic hypotension, they got dizzy, they fell, they broke their arm. I'd rather have a sense going into it that this patient was an intermediate metabolizer. I'd rather have a sense knowing that there's a drug-drug interaction that's going to add to this exposure, and we're going to have much greater exposure in this patient, and I want to avoid that. I mean, that's the whole point. So, get the patient on the right medication and the right dose early on in therapy. So I've been looking at medication lists since 1984, and I think back to all the patients I've seen that have had problems with their medication, and some of them, we never got right. And I just think, boy, what if I would have had this tool earlier on? How could I have thought about this mechanistically? Even when there weren't guidelines, how would I have thought about this? So we've got the drug-gene interaction. Again, metoprolol is at 2D6, star 4, star 17, the intermediate metabolizer. We've got the drug-drug interaction, metoprolol, paroxetine, changing that individual to a poor metabolizer. So it's a drug-drug gene interaction and phenoconversion. So this is not something that is, extremely rare. It's something that we need to be cognizant of all the time. So in this patient, we thought they were intermediate metabolizers based on the genetics, but in reality, they were a poor metabolizer. And now with 3 times what the normal metabolizer exposure would be, and you can see what the consequences were. Okay. So when we look at the guidelines then, how does this help us? Well, this is from the Dutch group, and I mentioned that we would look at this. And if the individual was a intermediate metabolizer without any context of the drug-drug interaction on top of the drug-gene interaction, the prescribing recommendation is no more than 50% of the standard dose. However, we recognize with the drug-drug interaction, the patient was phenoconverted to a poor metabolizer, and the guidelines say prescribe no more than 25% of the standard dose. So you can see the implications here as far as prescribing information up front. If I've got a patient who is an intermediate metabolizer, who is going to get a drug that might interact, I can have some context by looking at these guidelines and appropriately treating the patient. Of course, monitoring all along. And there are many other influences, certainly on the drug concentration. Again, physiology, pathophysiology, and other issues. So is this something that's rare? It is, and I mentioned that a minute ago. These are a number of different studies from a review that was done. And I'll just point to the second line where we see a wide range of co-medications given to some 865 subjects that were studied. The GPM, I put the G in there to stand for genetically there are poor metabolizers. 4% were poor metabolizers. But when you add in the phenoconversion component, the ppm here, we see 27%. So 208 patients, something like 210, I can't remember what it was, but a significant number of patients, certainly more than a fourth, right, with phenoconversion. So their genetics, what is reported on a report, a test results report where the patient's phenotype's listed as the intermediate metabolizer or what have you, that only goes so far. And that's why we really need to have the patient's medication list. It's absolutely, utterly critical. So here's what I'm calling Phenoconversion Susceptibility. And we've got three different genes that we mentioned already, CYP2D6, 2C19, and 2C9. And when we talk about phenoconversion, we have to think about, well, who can really be phenoconverted, right? It's really the middle of the pack, if you will. Those extensive, or what we now call normal metabolizers, and the intermediate metabolizers can be moved up or down pretty readily. If you're already a poor metabolizer, you're really at the bottom, and adding an inhibitor isn't going to cause much of a change. So maybe in that context, it's not a drug interaction. So here we have, really the context would be for extensive or normal metabolizers and intermediate metabolizers. Also, rapid metabolizers have been shown to be knocked down some, but maybe not significantly enough. But certainly we have metabolism inducers also. The anti-epileptic drugs as a class pretty much are notorious for increasing metabolism. And so we can think about that. And this is across these different genes, except for 2C9, we don't see increased metabolism from the genetic side anyhow. So I've added to this previous slide and put in some inhibitors of drug metabolism. There's the Flockhart table from Indiana University, a real great resource, and I've got the link down at the bottom of the slide. But you have strong inhibitors, moderate inhibitors, and weak inhibitors. And our patient that we had paroxetine, a strong inhibitor. So he was an intermediate metabolizer. He was susceptible to phenoconversion because of the addition of that inhibitor. And it goes beyond just prescription medication. For instance, cimetidine, or the trade name Agena, will be available over the counter. Now, it's a weak inhibitor, but what are the contexts and what are the consequences, I should say, of an over-the-counter medication? So we need to be thinking about this all the time. So here's just definitions of interactions, the drug-drug interactions, drug-gene interaction, and drug-drug-gene interactions, or phenoconversions. I won't read those. You can have those as a resource, just to keep you thinking about this. And there was a study, I think now 5-6 years ago. Actually, I'm looking at the bottom. It was 2014. So this study had over 1,000 patients, and they looked at interaction predictions, and they had this broken down into a couple of different categories. The drug-gene interaction, which is what we talked about first, then the drug-drug interaction, which you see was a high percentage here, almost 65%, and then the drug-drug gene interaction, about 22%. So phenoconversion, again, in almost a fourth of the patients studied. And of course, the more genes that you consider, the more interactions you're going to find. And you see a comparison of that over 1,100 patient study, and then an earlier study, which is referenced at the bottom of the slide also. So the point is that we're seeing this regularly. Again, maybe I'm beating the drum too loud. Now, I don't know if I can beat it too loud. It's there. We have to recognize it. So one of the benefits that we have at Manchester is a great relationship with Agena Bioscience, and Dave Miller and the group at Agena was really instrumental in helping us move forward in our platform that we use in our lab. So we're a smaller academic institution. We don't have a CLIA-certified laboratory. It's not a revenue generator from that aspect, but our Master's of Science in Pharmacogenomics students, both the on-campus and then we bring the online students in, get experience with this. And we really moved in this path because it helped us decrease our costs, and also efficiency-wise, we're able to look at not only the genotyping, but also copy number variants readily. It's all in the same workflow. And so when we look at an example here, what I did was I looked at the Veridose 4 panel from Agena, and then you'll see in a little bit that I randomly chose one of almost 2,500 patients from a long-term care database that we have, and then tried to put this together again as another example. But we're going to look at CYP2C19 and CYP2D6. And so here's the variants that are listed, and of course, again, one from mom, one from dad, and then the interpretation of the genotype to the phenotype and giving us some metabolic capability, again, whether it's ultra-rapid, rapid, normal, intermediate, or poor metabolism. And we have that going with this relatively limited number of variants, and then also with CYP2D6, more variants tested. CYP2D6 is the most varied of the CYPs, more than 140 variants. And of course, based on frequency and what population you're using, you're going to want to look at different variants. But anyhow, a thorough panel here for our purposes. So here's part of a report, a results report that I got back from some patients, and we pulled a random long-term care patient and looked at their medication. They were taking tramadol, an analgesic. They were taking clopidogrel and metoprolol. So two of the three drugs that we talked a little bit about. At least metoprolol we talked a lot about. So from the genotype standpoint, CYP2C19, the normal metabolizer, wild type star1, and again, that's a false here. So we expect normal metabolism. We have an intermediate metabolizer for CYP2C9 from that standpoint, and for CYP2D6, a normal metabolizer also. So what would we expect here? For CYP2C19, normal metabolizer, clopidogrel is the substrate drug there, and because we expect normal conversion to the active form, again, clopidogrel is a prodrug, well, we expect it to have amplified efficacy. Tramadol, again, CYP2D6, again, normal metabolizer, so we're expecting analgesic efficacy. And metoprolol also, CYP2D6, normal efficacy expected there also. But when we looked at the patient's list further, we saw that on omeprazole, a CYP2C19 inhibitor, and bupropion, a CYP2D6 inhibitor, a strong inhibitor. So now we've got phenoconversion expectations of CYP2D6 being converted maybe to, again, phenotypically, that's what the P stands for here, to an intermediate or poor metabolizer from their genetically normal metabolizer. And now, tramadol being a prodrug also, not as much of it's going to be converted to the active form, and so now we risk inefficacy here, right? Clopidogrel, again, prodrug, not getting converted to the active form. Omeprazole being an inhibitor, decreased efficacy for clopidogrel. And metoprolol, again, now with CYP2D6, but an active drug, It doesn't have to be converted, but it needs to get out of the body, decreased metabolism because of the phenoconversion, greater exposure to potentially adverse drug events. And if we go to the guidelines, there are no recommendations from the Dutch group nor from CPIC. But what do? Well, we've got that information, so mechanistically we can expect that there might be an issue, and we really, really need to look at this from a mechanistic standpoint when the clinical outcomes data that's really supported in these guidelines is not available. So again, when we added these inhibitors, we end up with an issue of changes in kinetics in this case. And I showed you where tramadol didn't work. Let's look at CYP2C19 and see what happens here. So for clopidogrel, we expected normal metabolizers, but because of the genetics, they were really more like an intermediate metabolizer, or maybe a poor metabolizer, and in this case, it changes our recommendation altogether. So where we would have used clopidogrel before, now we might want to consider, or should consider based on data, in the acute coronary syndrome PCI population, change to an alternative medication. And this is an example of phenoconversion again. So this is from patient data as opposed to the illustrated case I gave. So I've got some takeaways here before I turn it over to Robin. When we think about phenoconversion, we want to think about pharmacogene and drug exposure. And we want to think about that from too much or too little exposure. Every patient is going to have a baseline genetic exposure. It's going to be either increased or decreased or normal. And that exposure is going to be related to whether or not a drug is going to work or not work, and whether or not a patient's at increased risk of adverse drug events. So we've got those drug-gene interactions inherently. Now you layer on drug-drug interactions, and you've got another issue, which is phenoconversion. And when we look at phenoconversion in a bigger picture, there are some cases where it doesn't matter what the patient's genetics are, it doesn't matter what medications they're getting, because they have other reasons why they're phenoconverted. Take the liver transplant patient, right? The patient's coming in for liver transplant, the liver does not work. So they're a poor metabolizer. And there is no drug phenoconversion, there is no genetic phenoconversion. You have to think about things differently. If they get a transplant, then they get a different genome in that liver, which is a major metabolizing organ. So now you need to put that together. So the bottom line is, don't forget about phenoconversion. Don't forget about drug-drug interactions layered on drug-gene interactions. And with that, I will go ahead and turn it over to Robin. So thank you very much.
Thank you, Dave, for this wonderful presentation. It's a hard act to follow, but I will do my best to show you a little bit about how the technology works that underlies this particular testing. I'm not going to show you the particular genetic workflow or the laboratory workflow here. I'm going to talk to you about our platform. So it's a benchtop analyzer. It's mass spectrometry based, so it's very accurate and very sensitive. And because it is a simple system with software, you don't need a degree in bioinformatics. You just need good laboratory practice skills and molecular skills. And this platform has been used up to now in pharmacogenetics, oncology, hereditary testing, and also in COVID-19 research nowadays. The beating heart of our system is actually the chip that you see here on the left side of the slide. These chips resemble a 96 well or a 384 well PCR plates, and they are spotted with these little dots on there, which are the matrix. And this matrix, only after you apply your reagents to it from your biochemistry workflow, becomes specific to the things you want to analyze. So every pad in principle can be spotted with something different, and we can analyze on this platform SNPs, insertions, deletions, translocations, copy number variations, and methylation. It's a very simple workflow. The biochemistry steps only uses thermal cyclers, and our platform fits very nicely in between the real-time PCR, which can do a 1 to 5 assays per reaction, or maybe a few more, and in between the next-gen sequencing, where you can do hundreds of thousands of markers. We are a targeted genotyping platform, we can do tens to hundreds of targets per sample, and you can run with one system tens to thousands of samples per day. The beautiful thing about this platform is that you don't have variants of unknown significance. You only look for what you're interested in. And as you could see from Dave Kisor's presentation, you can look at assays that have an effect on metabolism or on clearance of a drug, for instance. How does this really work? So what happens is once you spotted your products from the biochemistry workflow onto your chip, then a laser irradiates each individual pad on the chip. This takes about 2 to 5 seconds per sample. And what happens in step 2 is once the laser is hit, you can actually, you get a cloud of particles, matrix, as well as your single base extension products. And these are going to be flown into a tube, a vacuum tube with an electrical current on them. And just like as in a kind of an agarose gel, you start seeing small fragments flying faster, bigger fragments flying slower. And so once fragments hit the top of the figure on the left, they are being recorded. When they hit, that gives you the time of flight and that gives you the mass identification. But also it can calculate how much actually is being hit on that detector. And on the right side, you see a small section of a spectrum of all the different peaks. And here you can see that there are 2 peaks in the spectrum. One is considered the wild type peak, and the other one is considered the variant peak. And we don't only get exactly where the peaks are and what the peaks are. We also get to know whether there is a certain amount under the curve for each peak, so we can detect how much of a particular variant is available or present. And the way we do it on our platform is we design the extension primers. On the left side, you can see the blue lines with the as in there. We design our extension primers based on the length of between 15 and 30 bases, and we incorporate a particular nucleotide in there. It's a termination nucleotide, so it only adds a T, a G, an A, or a C. And based on the individual weights of these nucleotides, we can then calculate upfront already what the peak masses are for each product in our spectrum. And then we can already, in this spectrum here, identify upfront exactly and during the time of flight which peaks are being present and how much of each peaks. So you can see in this case, this is the primer itself. The UAP KRAS on the left side, it's where the primer was. And on the right side, you have 4 different colored peaks or 4 different colored lines that go to 2 peaks there. Those are the possible extension products. So there's 2 things you can see here. One is that all the primer is being consumed and extended into a C and a G peak. And you can also, from this data, see that the C peak is about 30% and the G peak is about 70%. So it's not only qualitative, it's also quantitative. So in summary, the MassARRAY system gives you less weighting, more answers, very targeted results. It gives you the data that you need for the right medication, for the right patients, as Dave Kisor said so nicely. And having said this, I would like to hand it over back to Elizabeth for the questions and answers section.
Thank you very much, Robin. Yes, we have had quite a few questions come in. I'm not sure we'll be able to address them all, but we'll give them a go. Our first question is for you, Dave, from Yusuf. Will the degree of CYP2D6 inhibition by paroxetine be different if the patient's CYP2D6 normal metabolizer versus IM?
Yeah, that's a great question. It really does depend on the metabolizer status. I would say normal intermediate metabolizers can move down more so. It's a very potent inhibitor. The guidelines that are in place provide some information about that, and if you can go to the supplementary information about this, you can look at the resources that will provide the parent to metabolite ratios of the drug and the metabolites, so which I just said, and you can look at that, but it's not that well borne out in any given individual. There's more of a continuum with CYP2D6, and to the point, I don't want to get too much into to the point where we use something called an activity score. And so we're seeing activity scores being used also for CYP2C9. So there are differences there, but you have to think about the potency. And one of the resources, as I mentioned earlier, is IU Med's table of drug interactions, and they really do a nice job at defining what a potent, intermediate, and weak inhibitor are. I should say major inhibitor. It's based again on exposure and doubling or so at least some multiple of the area under the curve decrease in clearance.
Perfect. Thank you. Our next question, for a drug-drug gene interaction, does the exposure change over time?
Yeah, that's a great point. So when you change the metabolism of the drug, from a kinetic standpoint, you change the clearance of the medication, and the clearance will affect the half-life, how long it takes for the drug concentration to decrease by 50%. Think about it this way. If you have a drug that inhibits metabolism, the half-life is going to be longer. The drug's going to hang around for a longer period of time. That adds to that area under the curve, not just with higher concentration, but the fact the drug's hanging around for a longer period. To turn that around, if you give an inducer, so metabolism is increased, clearance is increased, the area under the curve is lower, the exposure is lower, like you would see in an ultra-active metabolizer, but also the half-life now is shorter. The drug gets out of the body quicker. And so that has implications in the dosing regimen design when we're talking about the dosing interval. So it also impacts so clearance will impact the maintenance dose, clearance impacts the half-life, and the half-life impacts the dosing interval.
Thank you. Darren asks, how would hepatic failure influence the interpretation of drug exposure relative to genotype, phenotype?
Yeah, so my first position was at the liver transplant for Presbyterian University Hospital in Pittsburgh, and we didn't have any of this kind of technology back then. We had to just look at patient responses, and I can remember, having patients who were getting 0.5 milligram of diazepam, and they were knocked out for 3 days. We have to really think about the phenoconversions that occur in that pathophysiology. It's liver-based. It's not really indicative of the genotype or related phenotype information. So that's where we run into problems. Now, in the study that we have ongoing right now, where we're looking at the genomes in the liver recipient and the genome from the patient, the donor, what we're doing is getting a piece of liver tissue before it goes in the patient, because what's in the patient, it's more difficult to get to. But what we're doing then is combining this information and looking at an overall phenotype for that patient. So we'll see. We're in the middle of that. We'll be looking as a second step at parent compound and metabolite ratios to see how the combination of the recipient and the donor genetics comes into play. But again, if somebody has severe liver disease to the point where they need a transplant, you're looking at the likely case of poor metabolism across the board.
Thank you. This question is from John. Is there a database that clinicians can use that will tell them which drugs, supplements, etc., are strong, moderate, or weak inhibitors of the various CYP450 pathways?
Yeah, so from the drug standpoint, again, that Indiana University website, the Flockhart table, David Flockhart, who is a clinical pharmacologist there, who actually passed away, but he really was an amazing leader in this area. So that's one really good, freely available resource. There's also a number of different online drug interaction databases that will tell whether or not there's a moderate or severe drug interaction relative to inhibition of different cytochrome P450s.
Excellent. This question is for Robin. Is the MassARRAY also able to do things other than pharmacogenetics?
That's a great question. Yes, definitely. The MassARRAY is in use for a lot of different things. Pharmacogenetics, of course, is one of the main components, but we have a very broad base in oncology, also hereditary genetics, and more recently, COVID-19 testing.
Thank you. One more question for Dave from USEF. phenoconversion can be tricky because the phenoconverted drug is also being metabolized by a different gene that could be polymorphic. So how do you reconcile all that information?
Algorithms that are developed. I mean, there's so much going on, right? Because you can have autoinduction too. I mentioned earlier that as a class of drugs, the antiepileptic drugs are inducers. Take carbamazepine as an example. You may start out with standard dose and have to quadruple the dose by the time the patient's induced. So they went from a normal or maybe a rapid to an ultra-rapid metabolizer. So that's another kind of case, right? CYP2D6 inhibitors, they can inhibit their own metabolism also. But then you've got the combination that you mentioned. That's not borne out in any guidelines right now, but you do have to think mechanistically, right? I mean, you also have to think about metabolite metabolism, if you will, right? So a prodrug like clopidogrel being converted to its active form, the prodrug has got a half-life of around 6 hours. The active form, I think, maybe less than an hour. So yeah, it gets very, very difficult. But from the standpoint of where we're at right now, taking in phenoconversion has to be at least addressed and considered when you're looking at a patient. There's no doubt that there's drug-drug interactions that layer upon drug-gene interactions. It's a first step.
All right, thank you so much. We are coming up on the hour, so I'd like to thank again our presenters for today, Dr. David Kisor and Dr. Robin Everts. I'd like to thank Agena Bioscience for sponsoring today's event. And finally, I'd like to thank those of you who came and spent this time with us. We're living in very strange times. We know you have a lot of choices about where to be, and we're very glad you chose to be with us this afternoon. So on behalf of BioIT World's Global Web Symposia Series, thank you all so very much for attending, and have a wonderful rest of your day. Bye-bye.